ako galibog ko na itong una. But don't worry, we will be discussing each and every process to be discussed in this video step by step aron makasabot gitang tanan. Good day everyone! I am Galleposo, and in this video, I will be discussing to you 3 topics related to nucleic acids. First, we have replication of DNA molecules, followed by chromosomes, and lastly, we have an overview of protein synthesis. Now, let's start with the first one, which is replication of DNA molecules. As we know, the structure of a DNA molecule is composed of 2 strands, which together form a double helix. And during DNA replication, these strands serve as the templates for the synthesis of 2 new daughter DNA molecules. But before we go any further, let us first clear up any clarifications. What exactly is DNA replication? DNA replication is the biochemical process by which DNA molecules produce exact duplicates of themselves. The result of DNA replication is the production of 2 daughter DNA molecules, both of which are identical to the one parent DNA molecule from which they were formed. So now that we know what DNA replication is, this leads us to our second question. How exactly does DNA replication occur? Well, it involves 3 simple steps. First, DNA helicase causes the DNA double helix to unwind, breaking the hydrogen bonds between the complementary bases. Now, DNA helicase is actually an enzyme. So as you can see in this picture, kaning DNA helicase, its mechanism can be compared to that of scissors. Pag gunting nimo sa papel, matunga siya. Maupod na ang trabaho sa DNA helicase. Iyang guntingon ang hydrogen bonds between the complementary bases aron dili na sila connected. Now, one thing we need to remember is the replication fork. The replication fork is the point at which the DNA double helix is unwinding, which is, of course, constantly changing or moving. Now, what does this mean? Diba ang DNA helicase, mumana siya ang maggunting sa hydrogen bonds between the complementary bases? Ang replication fork, mauna ang point kung asa nadapit gagunting si DNA helicase. And since DNA helicase is constantly breaking the hydrogen bonds between the complementary bases, the replication fork is also constantly changing or moving. In short, kung asa sa DNA helicase, diha pod nimo makita ang replication fork. Now, let us move on to the second step of the DNA replication process. The bases of the separated strands pair with free individual nucleotides present in the cell's nucleus. The pairing process occurs 1 nucleotide at a time. So in the cell's nucleus kuno, we have free individual nucleotides nga wala pa'y ka-pair nga complementary base. As you can see in the picture, ang mga free nucleotides kay nangadto sa unwinding DNA molecule and nagig-pair sa bases sa separated strands. Remember, adenine pairs with thymine and cytosine pairs with guanine. Now for the last step of the DNA replication process, after a free nucleotide has formed hydrogen bonds with the base of the old strand or template, DNA polymerase, also an enzyme, verifies that the base pairing is correct. So kaning DNA polymerase, mo ni siya ang tig-check kung sakto ra ba ang base pairing nga ni occur. If adenine paired with thymine and if cytosine paired with guanine. Because when a newly added nucleotide does not form proper hydrogen bonds with a template strand, this destabilizes the DNA strand, alerting DNA polymerase that there is a mistake. If the base pairing is incorrect, DNA polymerase removes the incorrectly paired nucleotide and adds the correct nucleotide with a base that complements the base of the template strand. Afterwards, DNA polymerase then catalyzes the formation of a new phosphodiester linkage between the nucleotide and the growing strand. So as you can see in this picture, we now have two new daughter DNA segments, both of which are identical to the parent DNA segment. As of now, we had just finished going through the DNA replication process. But, however, that was only an overview, in general, in a nutshell. Now, we will be going back and discuss the DNA replication process in finer detail. DNA polymerase can operate on a forming DNA daughter strand only in the 5' to 3' direction. Why? Well, in a nutshell, the nucleotide being added has 3 phosphate groups, as previously discussed. When it attaches to the DNA strand, 2 of these phosphates are removed, releasing energy. This energy is needed to form a strong bond between the new nucleotide and the DNA strand. If DNA polymerase tried to go in the 3' to 5' direction, it could not be able to use the same energy mechanism because the phosphate groups would no longer be in the right position. Without this energy, the enzyme could not be able to attach new nucleotides properly. In the 5' to 3' direction, if DNA polymerase finds an incorrect base pairing, it can remove the incorrect nucleotide using its proofreading function. But if it went in the 3' to 5' direction, it would not be able to fix the mistake since there would be no energy to continue the process because the necessary phosphate groups would already be gone. Now that we've already established that, let us move on. Because the 2 strands of parent DNA run in opposite directions, one is 5' to 3' and the other is 3' to 5', only one strand can grow continuously in the 5' to 3' direction. The other strand must be formed in short segments called Okazaki fragments as the DNA unwinds. The strand that grows continuously is called the leading strand, and the strand that is synthesized in small segments is called the lagging strand. Remember the recently discussed phosphodiester linkage between the nucleotide and the growing strand? This is what it's about. As you can see in the picture, the leading strand, kanang strand nga nasa taas, it grows continuously because it goes in the 5' to 3' direction, which is also the direction that DNA polymerase takes when it is verifying base pairings. However, the lagging strand, kanang ubos sa leading strand, it grows in segments called Okazaki fragments. Imagine unzipping a zipper and trying to sew 2 new pieces of fabric. On one side, the leading strand, you sew continuously in the direction that the zipper opens. However, on the other side, the lagging strand, you have to sew backwards in small sections because you can't move in the same direction as the zipper while keeping the stitching consistent. This mechanism is similar to that behind the lagging strand and the leading strand. The breaks or gaps in this daughter strand are called nicks, and to complete the formation of the strand, the Okazaki fragments are connected by the enzyme DNA ligase. Kaning DNA ligase, murang siya og glue nga mag-join together sa mga Okazaki fragments. Additionally, DNA replication is said to be bidirectional. Now, what does this mean? The process of DNA unwinding does not have to begin at the end of a DNA molecule. DNA replication is bidirectional for these locations. That is, it proceeds in both directions from the unwinding sites. As you can see in the picture, ang result ani nga multiple site replication process is the formation of bubbles of newly synthesized DNA. The bubbles grow larger and eventually coalesce, giving rise to 2 new daughter DNA molecules. This mechanism allows rapid replication of DNA molecules. As of now, we have just finished discussing the first topic of this video. But we still have 2 topics to go through. So now, we will be discussing about the second topic, which is all about chromosomes. Histones are proteins in the cell that provide the most stable arrangement for the long DNA molecules. Chromosomes are individual DNA molecules bound to a group of proteins, which are the histones. A chromosome is typically about 15% by mass DNA and 85% by mass protein. So as you can see in the picture, tag-as kaayo ang DNA molecules. So, kailangan sila ma-pack or ma-organize. And that's exactly what histones do. Ang DNA molecules wrap around the groups of histones aron ma-organize sila. And these DNA molecules and histones are what make up a chromosome. Now, just for a quick review, kay kabalo ko nga na-discuss na sa inyo in previous years, a normal human has 46 chromosomes per cell. Chromosomes occur in matched or homologous pairs. The 46 chromosomes of a human cell constitute 23 homologous pairs. One member of each homologous pair is derived from a chromosome inherited from the father, and the other is a copy of one of the chromosomes inherited from the mother. Homologous chromosomes have similar but not identical DNA-based sequences. Both code for the same traits but for different forms of the trait. And this is why siblings, despite having the same parents, look different. One sibling may have blue eyes while the other has green. They look alike but they don't look exactly the same. However, occasionally, identical twins are born. In such cases, the twins have received identical DNA from their parents. As of now, we have just officially finished discussing the second topic for this video. But before we move on to the last topic, let us first tackle about anti-metabolites, anti-cancer drugs that inhibit DNA synthesis. As we all know, cancer is a disease characterized by rapid, uncontrolled cell division. It is a deadly disease with many forms that has taken the lives of many within the past years. However, thanks to the advancement of science and technology, drugs that can be used to combat cancer have been developed. These drugs are called anti-metabolites. Anti-metabolites are a class of anti-cancer drugs that interfere with DNA replication because their structures are similar to molecules required for normal DNA replication. Instead nga ang real molecules needed for DNA synthesis ang magamit, ang mga anti-metabolites noon ang magamit ag sa similarity sa ilang structures. This trickery shuts down DNA synthesis, causing cells to die. Now, I will be introducing to you 4 commonly used anti-metabolites and the molecules they mimic. First on our list is 6-mercaptopurine or 6-MP. 6-MP structurally resembles adenine, leading to the incorporation of 6-MP rather than adenine into nucleotides. This results to non-functional DNA. So as you can see in the picture, similar kayog structures ang 6-MP ug adenine. The only difference is ang amino group or NH2 sa adenine is replaced by a sulfihydryl group or a thiol group in 6-MP. Next, we have thioguanine. Thioguanine structurally resembles guanine, leading to the incorporation of thioguanine rather than guanine into nucleotides. Again, the result is non-functional DNA. As you can see in the picture, similar kayog structures ang thioguanine ug guanine. The only difference is ang carbonyl group or C double bond O in the purine ring in guanine is replaced by a sulfihydryl group or thiol group in thioguanine. Next, we have 5-fluorouracil. The structure of uracil resembles that of thymine, which is methyluracil, leading to the incorporation of 5-fluorouracil rather than thymine into nucleotides. Again, non-functional DNA is the result. As you can see in the picture, similar kayog structures ang thymine ug 5-fluorouracil. The only difference is ang methyl group or CH3 in thymine is replaced by fluorine in 5-fluorouracil. Lastly, we have methotrexate. Ang previous three antimetabolites kay close structural analogs or na close structural similarity sa bases nga atong makita sa DNA nucleotides. However, methotrexate is not. Methotrexate structurally resembles folic acid or folate, which is one of the B vitamins. A derivative of folic acid, tetrahydrofolate or THF, is needed in one of the early steps of nucleotide synthesis. Methotrexate inhibits the conversion of folic acid to this needed derivative, shutting down DNA synthesis. As you can see in the picture, similar kayog structures ang folic acid ug methotrexate. However, they have two differences. First, ang hydrogen connected to a nitrogen atom in folic acid is replaced by a methyl group or CH3 in methotrexate. Second, ang OH group in folic acid is replaced by an amino group or NH2 in methotrexate. Now that we are done discussing about antimetabolites, we will now be moving on to the last topic for this video, which is overview of protein synthesis. Protein synthesis is the process by which cells build proteins, the essential molecules for cellular structure and function. The synthesis of proteins is under the direction of DNA molecules. It is this role of DNA that establishes the similarities between parents and offsprings that are regarded as hereditary characteristics. It occurs in two main phases. First is transcription, and second is translation. This diagram essentially summarizes the relationship between transcription and translation. But before discussing the protein synthesis process in detail, we first need to learn about RNA molecules as they are involved as the end products in transcription and as the starting materials in translation. This discussion will be brought to light by our next reporter.